The starting point for my talk today is a new buzzword that is making the rounds: convergence. Others call it biorevolution, nanobioconvergence, digital bioconvergence. At Catenion, we prefer the term bioconvergence. Whatever you call it, bioconvergence is one of those fuzzy concepts which are highly evocative but mean different things to different people. To become useful for biopharma executives, such buzzwords must be made actionable for company strategy, and this is precisely what I will attempt to do in this talk. My main contention is that we need to look at more than just technology to make sense out of bioconvergence. Technology is important, but so are the dynamics of the ecosystem within which innovation takes place. I first came across this concept a few years ago in the form of two MIT reports when working with the Israel Innovation Authority on a strategy to establish a sustainable biotech industry in the country. In 2011, MIT published The Third Revolution, the convergence of the life sciences, physical sciences, and engineering. And this was followed in 2016 by the report Convergence, the Future of Health. Three quotes from the more recent report clearly show that the authors had in mind primarily a technology-based definition. Convergence, as applied to health, is an approach to problem-solving that integrates expertise from life sciences with physical, mathematical, and computational sciences, as well as engineering, to form comprehensive frameworks that merge areas of knowledge from multiple fields to address specific challenges. The second quote reads, "Convergence is different from interdisciplinarity. Convergence is the integration of historically distinct disciplines and technologies into a unified whole that creates fundamentally new opportunities for life science and medical practice." And finally, today we are reaping the early benefits of a convergence revolution in which the tools, methods, concepts, and processes of engineering, including physical and computer science engineering, are increasingly used in biological research. Convergence in the domain of biology is of course nothing new. It goes back roughly 125 years when X-ray imaging techniques were first applied to biology. And 50 years later, Warren Weaver at the Rockefeller Foundation shifted the focus of the Natural Science Division from the study of inanimate matter to the application of techniques of physics, chemistry, and mathematics to the study of biological problems. The subsequent application of physical instruments like the ultracentrifuge, X-ray crystallography, electron diffraction, electrophoresis, spectroscopy, and electron microscopy to biology and chemistry ultimately led to the emergence of the science of microbiology. Today, an extreme version of bioconvergence postulates that not just health, but almost everything becomes intertwined with biology. Think of synthetic biology replacing textiles, plastics, meat, medicine, plants, and many other products with alternative proteins, and you will find platform companies like Ginkgo Bioworks, Novozymes, and others positioning themselves in this very broad space. Despite initial hype, it is still early days, and it remains unclear how useful this version is for biopharma. Focusing more on human health, the domain covered by the two MIT reports, the prevalent view of bioconvergence is less revolutionary. The main message seems to be that novel tools like machine learning, CRISPR, single-cell sequencing, and many others will accelerate current biopharma R&D processes and make them more efficient, leading to better and more predictive translational research and ultimately resulting in more effective precision medicine. A representative example of this view can be found in a recent article by Dr. Belén Garijo, the CEO of the German Merck. Vijay Pandey and Jorge Conde of Andreessen 16 envisage the emerging bioinnovation paradigm, although they do not call it bioconvergence, primarily as the application of engineering approaches to biology, where in principle, they say, every biological problem can be solved with the new convergent tools on hand. Biology, they say, is moving from serendipity to design. Now, personally, I can see a number of applications where novel tools and enhanced computing will help improve our biological understanding, but I have my doubts that we do and will have enough primary data to feed into those amazing tools and machines so they can unravel the complexity of stochastic multi-layer biological systems with their multiple inbuilt redundancies. In other words, the main issue seems to me to be that we tend to confound new tools to better manipulate parts of a system with an enhanced understanding of what, why, and how to manipulate in the first place. So I am sure we will see increasing process productivity of biopharma R&D, as we did indeed with omics and high throughput screening 20 years ago. Whether that will translate into improved output productivity in the foreseeable future is much less clear to me. So how do we at Catenion think about bioconvergence? Let me start by introducing the humble S-curve as a descriptor for biopharma innovation, where on the y-axis we measure the impact on disease burden and on the x-axis time. As with other fields of science and technology, we would expect a number of S-curves to follow and supersede each other over long periods of time. Although in biopharma, I would submit these curves do not intersect as they do in other industries. Why is this? Because in the biopharma field, innovation based on novel technology usually is more expensive than what it replaces. And no patient or physician or payer in their right mind would accept such an innovation if it showed an inferior efficacy or safety profile. Biopharma S-curves do not hang in the air. They can be characterized in terms of the scientific technology innovation space and the innovation ecosystem in which they develop. Now, we have described these two dimensions in quite some detail, but for our bioconvergence discussion today, let me just focus on a few highlights. And I'm, of course, happy to share more details in case you're interested. Starting with the scientific technology innovation space on the first S-curve, lasting approximately from the 1930s to the 1970s, we basically developed symptomatic treatments, mostly in the form of small molecules, with relatively poor PK/PD profiles and CYP interactions for large heterogeneous populations, all based on phenotypic screening. A second S-curve began to emerge in the late 1970s with the invention of hybridoma technology and recombinant DNA, followed by the various omics disciplines. More modalities became available, chiefly among them antibodies, and in what came to be called precision medicine, disease-modifying rather than just symptom-relieving drugs were increasingly developed based on target-driven discovery and targeting smaller, more homogeneous populations. Today, after 40 years, biopharma innovation has entered the rapidly accelerating part of this second S-curve, and it would seem to have way to go. But there are first signs of change triggered by the many novel tools we have previously mentioned. The innovation dynamics tend to move towards curative treatments for even smaller populations and often targeting individuals in a multitude of novel modalities, with the scientific approach increasingly characterized by co-innovation based on biological, mechanistic, electrophysiological understanding across all biological layers. In my mind, it is useful to think of this emerging S-curve as depicting the bioconvergence innovation system gradually replacing the biotech innovation system that we have grown accustomed to. Let's briefly look at a few examples of products and platforms rather than tools that might be classified under the umbrella of bioconvergence and which significantly increase and change the scope of traditional biopharma modalities. Note that many of these products and platforms are still in the early stages of development, and some, of course, may turn out to never work. Furthest advanced with a number of approvals are cell and gene therapies, oligonucleotides like the RNA vaccines and exosomes. They all require engineering, material science, new devices and data processing to develop non-immunogenic delivery vehicles with desired cell tropism to minimize safety concerns. Efficient logistics, CMC and quality control approaches need to be developed for drug development and will hopefully reduce unit costs so they become affordable. Next on the list are multiple digital health applications, a field driven by VC-funded startups, with both Novartis and Merck recently setting up incubators in this field. Then there's a multitude of emerging platforms at earlier stages, among them regenerative medicine, including the bioprinting of organoids, the creation of replacement organs like the bioartificial pancreas projects by Vertex, ViaCyte, Unicyte, as well as multiple wound healing applications. Then there's bioelectronics with the stimulation of the vagus nerve to control inflammation, pioneered by Kevin Tracey. This approach also applies to the splenic nerve, as well as medulla stimulation to control epilepsy and Parkinson motor symptoms. Brain-machine interfaces, originally developed by the Nicolelis lab at Duke University for reactivating motor control of paraplegics, and now leveraged by others, among them Neuralink and Sumitomo Pharma in collaboration with Melton, for reactivating lost arm and hand movements of stroke victims. Optoelectronics, optogenetics, originally developed by the Diceroth lab at Stanford to investigate the molecular biology of schizophrenia, and now in development to restore vision to blind people with degenerative eye diseases such as retinitis pigmentosa. And there are many, many more. For example, the field of biomaterials and the most astonishing work by Professor Michael Levin at Tufts on the bioelectric mechanisms behind developmental biology and their potential revolutionary impact on limb regeneration, cancer, and aging, which I discovered just a few months ago. All of these, and in particular Levin's work, clearly fit the bill of bioengineering based on artificial intelligence-driven insights into basic biology. While cell and gene therapies and digital approaches have by now become largely internalized into the biopharma space, the other examples listed above are happening largely unnoticed by biopharma in a parallel world, much closer to surgery and medical devices than to drugs. As we can see with regenerative medicine, clinical development, product approvals and launches, as well as pricing and reimbursement of these novel approaches are likely to follow completely different rules and dynamics to what biopharma is used to. So far, so good. Where it gets really interesting and where bioconvergence as a concept becomes relevant for biopharma strategy beyond which technologies to bet on is when we are looking at the second dimension characterizing these waves of biopharma innovation, which are illustrated by the S-curve. I'm talking about the ecosystem in which technological innovation takes place. Here the relevant questions are: who finances innovation, what type of organizations actually practice it, and what are the rules and regulations that determine how value is captured? It's probably a good idea to start this part of the discussion with the ecosystem which we all have become used to since the early 1980s. In a nutshell, biopharma innovation takes place in a free market environment with strong patent protection where shareholder value reigns supreme. It is financed by VCs and their investors at the earlier applied R&D stages and reinvested profits from blockbuster sales by established pharma companies at the later development stages, underpinned by $40 billion of annual NIH investment in biological research and a safety net of rapidly increasing net price levels, especially since the early 2000s. The characteristic organizations actually practicing innovation are the VC-funded startups and focused high-profit big pharma. Now the key question, of course, is this: was it always like that? And if it wasn't, then why would you assume the innovation ecosystem will not change again? So let's look back at the first S-curve, roughly covering the period of the 1930s to the 1970s. There were, of course, no VCs, no biotechs, no focused big pharma companies, and indeed no price inflation. Instead, innovation was driven by mid-sized companies, by today's standards at least, often subsidiaries of chemical conglomerates, operating in an environment of strong antitrust regulations, weak IP protection, and largely private insurance markets with low prices. Why did this change? If we look back to the discussion in the 1970s, it was dominated by how best to address a perceived drying up of innovation despite significant but decreasing investment of public funds into basic research via the NIH. Around 1980, the dominant political economy of Western countries radically changed. Remember the Thatcher-Reagan revolution, leading to major deregulation and the emergence of the current model dominated by VC-funded biotech and focused big pharma, all ultimately fueled by rapidly rising US price levels. If you are interested in the changes of rules and regulations that drove these changes, there are around 24 relevant ones to look at, prominent among them fundamental changes affecting IP, taxes, and investment. Innovation the policymakers wanted and innovation they got, but there are a few problems. The current biopharma innovation model is not fully leveraging the innovation potential of science, and it appears to be financially unsustainable. Now, first of all, biotech companies are not built to develop drugs or to last. They are essentially financial vehicles built for financial exit upon surrogate success, fueled by stock market speculation and open innovation models of big pharma. Too many good ideas in academia never reach the clinic for lack of funding and know-how. Too much investment is driven by hype, rising price levels and valuations, and comes to a halt when the business cycle moves into bear territory, as we are seeing right now. Economic incentives favor oncology and rare diseases, but the main disease burden of society in cardiovascular, metabolic, respiratory, and musculoskeletal diseases does not attract corresponding investment levels. As a senior policymaker at the Japanese Economics Ministry pointed out to me a few years ago, these factors taken together imply that the current ecosystem leads to an under-leveraging of the innovation potential inherent in science and technology, and thus effectively depressing the second S-curve. In the current debate in the US, the focus lies almost exclusively on increasing financial sustainability by controlling net prices, both among policymakers as well as some investors in emerging business models like EQRX. I see various problems with this narrative. Now, first, the quality-based approaches pioneered by NICE and now adopted by ICER in the US to evaluate how much we should pay for novel drugs may work for therapies that alleviate symptoms or deliver disease modification leading to limited impact on unmet need. But when we calculate with $100,000 or $150,000 per life year gained, they break down for curative treatments. And there can be, in my opinion, no value-based reimbursement approach to solve this problem. Secondly, it seems to me that rising price levels are not the problem. They are just a symptom of the underlying economic model of biopharma innovation, and in particular, how we finance translational research. What the future ecosystem will look like, we cannot know. But taking the current system for granted would be a grave mistake for sure. So it is legitimate, I think, to speculate, and I will offer some ideas to ponder. Let me begin by returning to the players practicing innovation. The predominant view today is that big pharma has enough firepower to acquire whatever external innovation it wants, and that will retain its role as the key player in the system. But this is an inside-out view of pharma, where established biopharma companies simply partner with third parties and gradually internalize novel technologies into their current business model. I maintain there may be more to it. Witness the multiple difficulties of established players with autologous CAR-Ts, the emergence of giant cell and gene therapies, CDMOs, and biotech-like deal terms for artificial intelligence and machine learning-driven service providers. An alternative view starts with players in other industries positioning themselves to take the lead in cross-industry collaborations. These include medtech companies like Fresenius and Medtronic, healthcare providers like Sheba in Tel Aviv or MD Anderson, big data tech companies like Apple, Alphabet, Verily, Microsoft, and multiple site entrants, often Japanese and Korean multi-technology conglomerates of the type that predominated in the US until the 1980s. Among them Fujifilm, Japan Synthetic Rubber, SK, Samsung, Lotte, and many others. As a consequence, it can be argued that in the emerging bioconvergence system, biopharma will be just one of different industry players that need to co-innovate. Is there going to be a dominant player? I don't think so. Although pharma thinks it can buy what it wants, and the AI industry would certainly like to make you believe that the key players are going to be them. Bioconvergence also implies that price levels can be expected to converge, likely downwards. We see early signs of this in the struggles biopharma companies are facing when trying to obtain reimbursement for regenerative medicine therapies that compete with significantly less expensive surgeries and require novel procedure codes. Downward price convergence presents pharma with a classic catch-22 dilemma. where companies may well pass on innovation opportunities that will offer lower margins than what the industry is used to, while side entrants might find these margins highly attractive. Phrased differently, for the first time in the last 50 years, bioconvergence may actually produce superior treatments at lower cost, a normal pattern in just about any other industry. Taking the argument a step further, we can speculate that the best way to increase sustainability and reduce the need for rising net prices is to reduce the role of expensive risk capital in the early stages of biopharma or bioconvergence innovation. This could be achieved by significantly increasing the role of public grant-based financing approaches. For this, novel funding agencies should be designed that can offer an alternative to VC financing with rigorous grant-based project selection and funding. The key here would be to avoid centralized approaches and instead to set up several funding agencies with partially redundant focus areas to avoid bias. They would be funded by a mix of budget appropriations and bonds, and just have a look at the bond-financed California Institute of Regenerative Medicine, now in its second round, the Cancer Prevention Research Institute of Texas, or MassBio. They would be working with rigorously independent advisors and making funding decisions based on clear, transparent decision criteria prepared by professional decision analysis input on the model of NICE in England or ICER in the US. And they would be funding projects organized on the model of DARPA to be managed by experienced biomedical executives and young professionals on competitive salaries, receiving social recognition, but with limited financial upside. What would be the financial rationale for such an approach? By reducing financial risk of investors in higher risk ventures, the pressure on pricing of the few successful projects that make it through would be reduced. Established companies could be offered licensing agreements of proof of concept stage projects without large risk premiums and be asked to accept price points based on the health economic analysis by independent institutes like ICER. Ultimately, increased public investment into translational research would likely pay for itself via reduced prices and reimbursement for the resulting innovation. I will lay out the principles of a workable approach, how this could be organized in more detail in another video, where I will focus on regenerative medicine, which is the field for which the current system arguably works least well. Now, if you think this sounds too radical, then just look at the changes that happened around 1980. Bioconvergence, in this view, is just another wave of innovation based on a different set of technologies and bringing with it different rules of the game. What these rules of the game will look like, as we said before, is an open question, but longer-term company strategy should begin by reframing some of the basic assumptions underlying current business models. It seems clear that along with bioinformatics and machine learning and so on capabilities, companies will need to build engineering-like project management skills across multiple domains, capabilities, and players, and develop strong networking with the early-stage academic research community beyond the field of biology to identify future trends. To come back to the points raised by Andreessen's Pande, I think we should not forget that engineering biology is full of counterfactuals. What I mean is different things can happen based on the same signaling. The reductionist view is that we simply need to fully understand and map all signaling pathways and their interdependencies. Another view is that this is simply not possible due to the stochasticity, plasticity, and different layers of biological processes, as well as the challenges of upscaling synthetic biology. In this view, the main challenge of bioconvergence will be to drive biology beyond reductionism, and at the same time, develop a culture and language to make it talk to engineering, which is reductionist by definition. And that is a tall order. And tackling this challenge could ultimately, I believe, provide significant competitive advantage to companies who master it. Culture eats strategy for breakfast, as the saying goes. Another question for strategy is whether cross-industry co-invention required by bioconvergence might be delivered more effectively by internalizing several parts of the puzzle into a multi-industry conglomerate rather than project-driven consortia of narrow specialists in multiple shapes and sizes. I have no little answer to this question, but my gut feeling is affirmative. So let me come to a conclusion now. First of all, bioconvergence is not just relevant for established biopharma companies and side entrants, but also for policymakers, as we have just seen when discussing the funding of translational R&D. The Israel Innovation Authority started with the question how to build a sustainable biotech industry. Leveraging some of the ideas outlined in this webinar and the country's strengths in biology, tech, engineering, nanotechnology, machine learning, and electronic longitudinal healthcare data, they ended up with bioconvergence as the next growth engine of Israel's economy. So what then is bioconvergence? I would say it is a convenient label for the next wave of biomedical innovation. Why? Because it describes the increasing scope and pace of co-innovation across disciplines and industries, convergence, that we are seeing on the technology side. And everybody can see that. My main point in this talk has been to draw your attention to the other side of the coin, namely the ecosystem within which innovation takes place. Who finances innovation? What types of organizations actually do practice it? And what are the rules and regulations that determine how value is captured? We've seen that the answers to these questions have radically changed around 1980, just as recombinant DNA and hybridoma technologies were beginning to drive biotech innovation. My contention is that we will likely see similar radical change over the next decade or so, and I would say that a solid strategy needs to incorporate such changes at the level of potential scenarios instead of taking the current ecosystem for granted and just focusing on which technologies to invest in next. The need for sustainability and the promise of better products at lower cost should drive the conversation going forward. Thank you.